much, Monica and Sanjana and this whole gang at IIMD for inviting me here. I must say that in recent years that the whole the attitude towards rare disease is changing, and I think ICMA is taking a leading role towards this area. So after Shefali talked so much about this thing and giving a really a great insight into the disease and various treatments, I'll just focus on a disease. When we started, it was absolutely rare. I hardly knew one or two patients in the whole country. And then last 10 years or so, it has gone far. And I'll talk to you about how one goes about developing a therapy towards a disease which is very, very rare in one of the muscular dystrophies. So let's start. Next slide, please. So essentially, just to give you a summary of what GNE myopathy is, that it's a recessive genetic disorder and it affects the bifunctional enzyme. Generally, it is called GNE. Yeah, so can you hear me now? Hello? So it's a bifunctional enzyme called GNE, and there's a mutation in this enzyme that causes, this enzyme is involved in sialic acid bisulfite. And unlike DMD or other, this affects all skeletal muscles and without touching actually all the smooth muscles. Even in skeletal muscles, it actually spares quadriceps. Very surprisingly, it spares quadriceps but does not, then affects all the skeletal muscles. It's a late onset disease, so it has problems that are different from the other ones. And generally the symptom starts around 20 and or 55. GNE is a rate limiting enzyme, and we'll talk about that, and involved in sialic pathway. In terms of therapy, we'll talk about it. Because it's a sialic acid biosynthesis, so sialic acid supplementation is thought to be one of the major therapeutic options that we have. So, but various trials that was carried out, there was a trial that went up to phase 3 for supplementation of sialic acid, and it failed at that time. The endpoint was not reached, and the trial was suspended at that point. NIH has carried out another trial, which is the kind of a precursor of sialic acid called N-acetylmanosamine, and they've concluded up to second phase and not been able to take up last 3-4 years to the phase 3 trial, because I think there's some problem of funding into that. Next slide. Next slide, please. In India, the first patient was diagnosed in 2006 in NIMHANS actually. And currently our record says that more than 300 people have been diagnosed. And there's only few places that expertise are available. Not many people see this actually. And DNA testing is the final diagnostic point. That's what it is. Muscle biopsies are not anymore done. Generally, people are avoiding doing muscle biopsies. And there has been an attempt to link mutations with the progression of the disease. There's no clear linkage of that. And then we also found it that India has a predominantly one mutation, and most of the people have compound heterozygous mutation in India. And one of the mutations is absolutely restricted to the Indian subcontinent and not seen anywhere else. Next slide. It is not that rare. If you look at some of this one pathogenic mutation that has been seen, its frequency can be very, very high. Among a small Gujarati population, we saw 14 to 15%. and whereas pan-India it's 1.5%. It's very common among the Indian people. So one can estimate roughly, and generally the estimate is in India about roughly 30,000 or so, and somebody from America did an estimate based on a Jewish mutation, and they also think that worldwide is something 50,000 to 1 lakh patients. Next slide. So as I said, the gene is actually involved in sialic acid. It's an absolutely essential, sugar molecule, and it's present in all tissues, brain, liver, everywhere, and almost all tissues actually expresses GNE gene. But the defect is only in the skeletal muscle, and that's what the major problem is. That do we understand what it caused the difference? And in fact, our study, I'm not showing you here. So we looked at, there are 7 isoforms of messenger RNA, and we found that muscle has only 1 isoform which other tissues don't have. So we suspect that the expression of that isoform is what is maybe affected, and that's what causing the problem. So this gene is essential. Non-mutations are absolutely lethal. Okay, so and nonsense mutation only in the heterozygous situations are seen in this case. So there are, in the pathway, there are many ways you can actually supplement, and we did a mathematical modeling once to see what it is. Sometimes the lower level sialic acid is supplemented by degradation process that are stimulated. But anyway, there are many ways you can introduce this. Increase the level of by supplementing at different levels. Next slide. What is the biology? As I said, that reduction of sialation of proteins and sialic acid level is thought to be the major cause of the disease. But there is an alternate mechanism that says that GNE gene also has a role in interacting with other proteins, particularly cytoskeleton proteins like alpha-actinin. And the reduction in that interaction can also cause diseases. So what is not known, as I said, genotype to phenotype, it's not clear why only muscle cells get defect, whereas the gene is expressed everywhere else. And that's the major cause of it. And as I said, that one of our hypotheses is that one isoform expression that happens only in muscle may be the one that may be responsible for it. Next slide. So what are the potential treatment? The 2 ways you can treat it. Either you treat it the defect that muscles are happening, try to increase the health of the muscles, or you actually treat directly the mutations that are happening, and gene editing, etc., the gene therapy, that can handle that. So we think that we should try both, and maybe a combination of both the treatment would be very useful for the disease. Next slide. So in a disease like this, a very slowly progressive disease, it's very important to have natural history of the disease, because you need to know how the progression of the disease is happening in patients. Shefali talked about progression of the, but in a slowly progression disease, if you're going to do a trial of a new molecule, then how much you are going to actually expect within 2 years of the trial cycle. So in order to actually decide your endpoint in clinical trials of such diseases, you need to have a natural history of Indian patients. And it's very, very important to do that. And we have actually, and because it generates a quantitative data about how the disease progression will happen. And that's very, very important to do that. And it has been done for some Japanese patients with a totally different mutation, some American patients, but obviously large number of Indian patients not done. As I said, Indian patients have different kinds of mutations. So we have actually initiated natural history study. Next slide. Next slide. So we have started natural history study with Nimrans, and our organization is supporting this study. And whole lot of this study is based on a couple of things, based on questionnaire, based on muscle strength, based on MRI muscles, based on blood and other urine samples. Tata Institute of Genetics and Society is involved in it. And we have people who can make Bayesian statistical models of progression. So already we have recruited a very large number of patients in it. And we plan to collect data from each patient every 6 months to evaluate. And the study is already underway in this. So we believe that this study will actually pave the way to do clinical trials in this case. Next slide. So the other possibility, as I said, is a metabolite replacement therapy. So what is going on around the world? Next slide. So one study, and this is something that patient groups should learn from. There is Michela is a patient of GNE myopathy living in Italy. She, in fact, collected scientists and wrote a project for European Union. There's something called PROTAC. That means it's a precursor of sialic acid, which actually can be, it is designed in a way that it will be transported. Sialic acid does not get transported into cells because it's an acid. So it's a modified form of sialic acid that gets transported into the cell and then get then modified there, remodified actually, and the sialic acid is generated. And this drug is what it is. So she was very successful getting money and getting a bunch of scientists, and they carried out a lot of study. They developed a zebrafish model and found that this drug is working. Now they finished the first part of the study, waiting for funds to continue the study. So this is one effort that how a patient and patient groups can actually initiate a research study. Next slide. The other study that has been completed and got approval from Japanese Japanese drug agency is the modified form of sialic acid. Again, it's a slow release modified form. And they carried out this study and they found that upper body shows a considerable improvement in this molecule. And so there is a company that taken up manufacturing, but they're not able to produce enough quantity even for our patient. I think some of these things we should be able to make in the country. But this is working. This is the potential molecule that it is. Again, it is a metabolite replacement. It's a modified form of sialic acid. Next slide. So there is another Korean effort in a modified form of sialic acid. This is sialyl lactose. Again, it is present in the human male. Actually, we get a lot of sialic acid through sialyl lactose. Our babies get sialic acid because they actually digest this and produce sialic acid. So this also restored sialic acid. Again, this has got approval in it. This molecule is actually available right now as a supplement, and one can buy it. You can get $199 per bottle for a 1 month supply of this molecule. And obviously we can make it even cheaper than that in this country. But then again, this is working. They are not great molecules, but they actually reduce the, as Shefali was saying, anything that reduces the progression is itself a good idea. Next slide. Autophagy activators are turning out to be good drugs for many of the muscle diseases, and many of these diseases, autophagy, mitophagy defect has been found. So any molecule that improves it can be done, and this I'm not going to detail in it. Autophagies are a mechanism by which the waste material or some of the materials are degraded and reutilized in the cell. So next slide. Many diseases, autophagy, mitophagy defect. Next slide. So and there are a number of molecules known for these cases, and so there are a large number of such molecules which have been approved already by FDA has been tried. in gene myopathy, it has been shown that one of these molecules actually works in a very good way in the cellular model system. It's been taken up further. Again, funding sometimes creates a problem for taking some of these things. So these are the potential molecules that can actually help in doing it and already showing potential improvement in it. In case of DMD mouse models, this autophagy seems to work. Next slide. There are different forms of gene therapy. Our organization is supporting some, and in some ways, in a very small way, we give very small support for people to initiate doing that. Next slide. Messenger RNA therapy. This is the project that Tata Institute of Genetics Society has initiated. They're developing a messenger RNA platform for 3 diseases: DMD, GMD, and Pompe disease. So they are also looking at DMD myopathy. We supported them initially to start the project, and essentially the idea is that, unlike enzyme replacement therapy, here what you're doing is that you're giving the protein using messenger RNA. Messenger RNA goes inside the cell and makes the protein, and that can be useful for almost any diseases that you, wherever the gene therapy is. Is it one form of gene therapy? There are a lot of advantages in this. The toxicity will be less, production cost will be much less, but you have to give it repeatedly. So your system should have very low immunity. So you make the messenger RNA, you can make it very well thanks to techniques developed because of the COVID vaccine, mRNA vaccine. You encase them in lipid nanoparticles and then you inject it. But unlike vaccine, you have to inject much more. So a little bit of toxicity issues are there, and people are designing good LNPs that would have reduced toxicity. So already there is work have been done where messenger RNA based systems have been produced. GNE myopathy is being expressed in cells, and also in Pompe disease it is being also shown to be expressing their cells. So it's a very potential thing. And already India has a manufacturing base. People who are making mRNA vaccines, they have a manufacturing base for this technology. Next. Non-viral vectors, Shefali mentioned it. This is the Munia Ganguly's laboratory is doing it. We supported them. And they also make a non-viral vector for GNE myopathy, and which seems to be expressing in it. They are taking it up further for the study. The advantage of it is that it is non-toxic and it's much cheaper, but you still now the level expression that you want is a little lower, and one needs to build more on that. More optimization is needed to do it. Next slide. Yes, I won't talk about it. Arta Subra will actually talk about this much more. But his group has all the systems ready. He's going to show you data on DMD, but GNE myopathy he's done some work. He has shown that it expresses. This is all ready to take it up for further research. He has a production system. And since the GNE gene is smaller, much smaller, so it can be accommodated in the vector more easily. You can accommodate the whole GNE gene into the AAV vector for this purposes. And we are looking forward to it and hope that he gets more funding so this can be actually taken further. The cost will be much more. They are also doing a lot of immunological study, which I know. Next slide. So there is a novel gene therapy that is coming up in this area where it's an interesting idea that using a vector that expresses 2 different proteins, you express the gene of interest like GNE, and you also express some other protein that increases the muscle health. Okay, in this case, cholestatin has been used. Okay, so by expressing both at the same time, you not only providing good gene but also increasing the muscle health. And this system has been developed. They are taking it further for GNE, and let's hope that they can do that. And this is happening in the USA. Next slide. Okay, so I sort of give you an idea about various kinds of therapy that is being developed for genome. Some of them are already available if we can make those molecules in India, or we can try to import them into this case. I would suggest that given our strength in chemistry, we should be able to make these molecules in India. As a patient organization, we took the initiative that we want to get these molecules made in India, developed in India, we think. So we have not done great. I mean, we give only very small grant, we give it to Arta, give it to Lumia, give it to Gates, just to initiate this study. I think patient organization could do that. Obviously, supporting the whole study is not possible. But at least get scientists interested in your project. If you get scientists interested in your project and they start working on it, someday you will be able to actually see. And you can see that gene hematology is really on the verge of, and therapies are available, and I think, I'm sure next few years we would have some of the gene therapies coming up. And thank you very much for giving the opportunity.